So this ribosome have two sites for tRNA. By this, using this codon, the anticodon will bind, particular complementary anticodon will bind with this codon. For example, here it is A, so U, A and C anticodon having tRNA will bind with this codon which have particular amino acid which code for the, it is a AUG, so it is code for the methionine. Okay, now what will happen after this? What another tRNA come bind with the another triplet codon. Here it is C, so it complementary base is G and U. Again, this tRNA have particular amino acid. Then after two amino acids come together and one polypeptide bond is formed. After this formation of peptide bond in amino acid, this tRNA is come out from the ribosome. Okay, so it is come out here. Now this amino acid is free. Now likewise another tRNA will come here, U, A and C having tRNA here have specific amino acid will again bind with this amino acid by peptide bond, and after joining with the by the peptide bond, this amino acid free by the tRNA, and this tRNA removed from the ribosome. Likewise, this process will continuously occur up to the terminator codon, when the ribosome reach to the terminator codon, this process called translation, and ribosome now separate from this mRNA, and this polypeptide chain now it is known as protein, which is separate from the tRNA and then go for their further work in a cell. Now what happened with this tRNA? After formation of protein, this tRNA will degrade into the cell by the cell component itself. So this is the process known as translation. In a translation, RNA produce protein, particular kind of protein on the basis of which kind of codes are present on tRNA. This is mRNA. Proteins will form on the basis of codes that are present on mRNA. What is the function of tRNA? tRNA provide the amino acid to the mRNA strand. Okay. Now likewise, this amino acid bind with the peptide bond, and at the end one protein will form. This process is known as translation. Here translation, and this whole process is known as central dogma of molecular biology. Means the molecular biology is everything about duplication, transcription and translation. So why these processes are so much importance in the life, and why we are studying these processes? Because how DNA expressed in our cell is most important in the genetic disorder, to finding this genetic disorder and for the cure of them, this kind of disease. For example, there is one disease known as cancer. What happened in cancer? cell continuously grow. We know that cell continuously grow. What it require? It require continuously duplication of their genome or their DNA. So in cancer cell, what it require? It require continuous production of the replication, continuous process of replication in which DNA makes copies of themselves. There are so many copies of this cancer cell produce so many copies of DNA, and this DNA goes into different cell and form lots of cell, abnormal division of cell and produce cancerous cell. So how can we stop this cell, production of cell or division of cell? We know that in replication there is one enzyme called as a telomerase enzyme. Which is responsible for the telomere sequence. Telomerase enzyme give the telomere to the replicating DNA. If we remove this telomerase enzyme from the cell, what will happen? They don't have, this cell don't have a telomerase. So this cell division or duplication of RNA will not, duplication of DNA will not synthesize, means this DNA will not synthesize, and the further growth of those cells will arrested by the removing of telomerase enzyme. The function of telomerase enzyme we will discuss in a higher classes. Now this is about the how DNA expressed in our bodies, and this process or this theory known as central dogma of molecular biology. Now we will discuss about, in next class we will discuss about the Human Genome Project and how this expression of protein and expression of gene take place in our body or in an organism. Thereafter we will discuss about Human Genome Project, that what is Human Genome Project and what are the goals of Human Genome Project, and then we will discuss about DNA fingerprinting. DNA fingerprinting. So what is DNA fingerprinting? This all we will discuss about in a next class. Okay, thank you.